exosomes is a specific type of extracellular vesicles. So these are small containers that are secreted by cells, and they contain a group of biomolecules, lipids, proteins, and also genetic information, RNA, for example. And these containers of biomolecules, they are sent to target cells, and target cells receive these containers and as kind of messages, and the messages will of course influence then the behavior of the target cells and modulate their kind of biology. So this is a kind of communication process that can happen in the brain. And as we know, the brain is very much built on interaction between cells, and it's not only neurons that interact with each other, it's also glial cells and neurons that speak to each other. And so this is a kind of a new mode of communication that has been established now in the brain, that cells send these kind of containers of biomolecules, and there is a kind of crosstalk between the cells and influencing each other. But our work focuses actually on communication between glial cells and neurons. So we discovered that oligodendrocytes, which have a close, intimate relationship by forming the myelin sheath with neurons, they secrete this kind of exosomes. So these exosomes actually is a specific subtype of extracellular vesicles, has a very selected cargo. And so oligodendrocytes send these vesicles to neurons, and the cargo is made available to the neurons, and it seems that neurons feel more happy after they have received these packages, and that's why we call them also care packages. Neurons are very specialized cells, so they have a very special architecture, sending these long axons, and so they are made for firing electrical signals, or generating electrical signals. And this is very energy consuming, and they have to maintain their architecture. So, and they need some support from outside to maintain the long-term homeostasis. So, and we think this is the process where these packages are important to support these cells. And that's actually our working hypothesis and our first data point to that idea that we are right. But there's, of course, a lot of more work to be done. We have some models of degeneration, actually, where we think this kind of support is lacking. So, for example, demyelinating diseases, or in some genetic models where some glial molecules are missing that are part of the signaling pathway. In these models, the axons actually degenerate. So this kind of support seems to lack in these models. So there, it of course turns at some point into pathology, but we are using the pathology to understand the physiology behind it.